Thank you very much. It's really a pleasure to be here today and be able to tell you about some of the work that we've been doing. So just an outline of what I'm going to tell you about today. First, I'll tell you about gene therapy in very general terms. Then I'll tell you specifically about one particular gene delivery vehicle, adeno-associated virus. I'll tell you about a lot of choices we get to make to help us determine the control of expression or where the transgene we're trying to deliver, where that ultimately winds up being expressed. I'll show you the different Barth syndrome vectors that we've developed, and then I'll show you some preliminary data. So first of all, gene therapy is basically the delivery of a healthy copy of a gene into cells with a missing or defective copy to treat a genetic disorder. It's not, or one thing to remember is that it's not really a one-size-fits-all type of therapy, so each gene therapy must be really customized to address the distinct features of the disorder that you're trying to treat. And I'll show you a bunch of different things we're doing to specifically target Barth syndrome. So in developing a gene therapy for Barth syndrome, these are a few things that we really need to keep in mind as we're developing these vectors. One thing is we really want long-term expression. We would like for this gene to express for the duration of the patient's life if possible, or at least as long as possible. We'd like to target heart and skeletal muscle, and that doesn't have to be written in stone. If you tell me another tissue needs to be targeted, we can adjust this at some point. We want to determine the minimal effective dose, and this is not really just for gene therapy, but any therapeutic, whether it's a drug or anything else, you want to figure out what the minimal effective dose is. This is important for a lot of reasons. One is because if there's less drug or therapeutic that you deliver to a patient, you're exposing them to less potential for side effects and other problems. The other thing is if you can use a smaller amount of whatever you have available, then perhaps you can treat 40 patients instead of 20 patients, and it's just more efficient. So in developing the gene therapy vectors, there are little tricks that I'll show you we can do to try to think about how can we get the most bang for our buck from our therapeutic vector. We're also interested in always thinking about how can we minimize the potential for any sort of immune response. And then patient safety, it's last on this list, but that doesn't mean anything. It's really number one. We want to make sure that every step of the way we're really thinking about patient safety. So with regard to gene delivery vehicles, there are a lot of options out there. There are lots of different plasmid delivery systems. There are retroviruses, lentiviruses, adenoviruses, and adeno-associated viruses, which is what I'm going to tell you about today. All of these have different strengths and weaknesses, and there are different reasons you might want to use one versus another depending on what your application is, and I'd be happy to talk to any of you later about why you would or wouldn't want to use various ones of these for different reasons. But today I'm going to really tell you about adeno-associated virus, which my colleagues and I feel very strongly is probably, given the different options of gene delivery vehicles that are available right now, this is the most promising one. So a little background on adeno-associated virus, or AAV. It was discovered as a contaminant in adenovirus preparations, hence the sort of strange name adeno-associated virus. And it's non-pathogenic. If I'm talking to real virologists, you know, like people who study Ebola and things like that, they think this is a pretty wimpy virus because even after it enters its host, it can't replicate on its own. It really requires a co-infection with adenovirus or herpes virus or something else to help it reproduce. So it's, in the sense of real virology, it's not a very impressive virus, but from a gene therapy perspective, that's kind of a good thing, that it doesn't replicate very well on its own. It makes it safer. Furthermore, the recombinant AAV, or rAAV, that we're using in all gene therapy vectors completely lacks the elements necessary for replication that are provided by AAV normally. So if you look at the top example, that's wild-type AAV. Oh, I'm sorry. Uh-oh. I'm so sorry. Oh, phew, okay. So if you look at the top example, you see these ITRs, and that stands for inverted terminal repeats. Those flank a few replication genes and a few capsid genes. And the total capacity of this is about 4.7 kilobases. If you look at the bottom example, that's recombinant AAV that we're using in gene therapy. And so the rep genes, the cap genes have been removed, and they've been replaced with a promoter driving expression of whatever transgene you're interested in expressing. They still sit between those ITRs, and in the end, in gene therapy vectors, the only bit of viral genome left is those inverted terminal repeats, and those are necessary for packaging into the capsid. So again, AAV is non-pathogenic compared to with other viral vectors. It elicits a very weak inflammatory response. You have all been exposed to various serotypes of AAV in your lifetime, but you had no idea that it happened because it had no effect on you whatsoever. So another interesting thing about AAV is that it persists as an episome within the nucleus, and so it's stable for many years in vivo in non-dividing cells, but eventually it becomes diluted out in dividing cells. And so this picture down here is sort of showing you AAV enters. It's a single-stranded DNA virus, so you see the single strands, they become double-stranded, and then over time they persist either as these little episomes that sort of look like little plasmids. It also can form dimers or concatemers, but the really important thing is that integration into the genome is extremely rare, and so that makes this more safe. Also, you don't have to worry about if we deliver this and it enters into the genome in a random spot, you're not going to accidentally knock out some other gene by trying to develop a therapy for Barth syndrome. Another key characteristic about AAV is that many natural and various research-engineered serotype capsid options exist. So what this basically means is even within the constraints of just AAV as a gene delivery vehicle, there's a whole parking lot full of different vehicles that are options that you can choose. And so different AAV capsids have different tissue tropisms. So you think about what tissues you're trying to target, and you choose a capsid that makes sense for your target. The one big thing that most people feel is a limitation of AAV is its limited packaging capacity size. So it's 4.7 kilobases of what it can fit as far as coding sequence plus your promoter. We're very lucky. TAS is a relatively small coding sequence, so this isn't a problem at all for Barth syndrome. So there are many levels that you can look at to try to control expression and really restrict it to the tissues that you're most trying to target. One thing you can choose, as I mentioned, the capsid serotype, and I'll go into more detail about that in a minute. You can also play around with your vector sequence design. So you've got choices you can make in different promoter elements. You can choose tissue-restrictive promoters. You can perform codon optimization. You can use double-stranded vectors, and I'll show you some examples in a minute. You can also make decisions about your delivery route, depending on what you're targeting. So with regard to different capsid serotypes, a long time ago when I was a graduate student in Barry Burns' lab, we did this project where we were comparing lots of different AAV serotype capsids and looking at where do they just naturally go, what is their natural tropism. And we really found that AAV9, which is a natural serotype, was on its own, just very cardiotropic. That doesn't mean it doesn't go to other tissues, it just really likes the heart. So another level of control you have is your choice in promoter usage. So tissue-specific promoters will increase expression specificity and help restrict it to the tissues you're interested in. And we did another study a while back that we were comparing a lot of different promoters that we suspected would work well in the heart. And we were interested in those that would work well only in the heart, as well as trying to find something that would work well in both heart and skeletal muscle, which is something that would be useful for Barth syndrome. And we found that the Desmond promoter really does a good job of transducing both heart and skeletal muscle. And so this kind of goes back to the thing I was mentioning before about the minimum effective dose. So if I can give more bang for my buck for each virion that I'm delivering, if I can make it express better, I will. And so one way you can do this is through codon optimization. And so many of you are aware that 3 nucleic acids are required to encode an amino acid, and so that's a codon. But amino acids can be encoded by more than one codon, and different species have preferences for different codons. They're slightly more efficient in different species. So you can go to some online sites, and there are these algorithms where you can just plug in your amino acid sequence and say, I want this codon optimized for human or mouse or whatever you're interested in studying. And then people have found that if you use something codon optimized for the species that you're actually using, you'll achieve faster translation rates. So this is an example of what a clinical vector could look like for Barth syndrome, and in fact, we've cloned this and we've got it ready. So you can see it's the ITRs are flanking what we're delivering, which is in this example, it's the Desmond promoter driving a codon-optimized version of the human TAS gene, and then there's a poly-A tail. And so, importantly, the total size of this is really only about 2.4 kilobases. So our promoter plus the tafazin gene, it's well beneath the constraints of AAV. AAV can hold about 4.7 kilobases. So this is the perfect length for a double-stranded vector, because you do want to get up as close to that 4.7 as possible. That helps with packaging efficiency in AAV. And so we get to use double-stranded vectors, and this is really the perfect size for this. And I'm not going to go into this in great detail, but I'll just tell you that if you look on the left-hand side, that's what normally happens. In the end, you have this single-stranded DNA, AAV DNA, flanked by these ITRs, and that enters the capsid. What we can do by using double-stranded vectors is use a vector that has a mutation in one of its ITRs so that those no longer become resolved. And in the end, you sort of have this dimer that then enters the capsid. And so one rate-limiting step to expression of AAV once it gets into a cell or a tissue is that you can't express anything off of single-stranded DNA. So second-strand synthesis has to happen, or it needs to anneal to a similar strand of DNA, something like that needs to happen. And to sort of make this more efficient, if you're delivering this dimer, it simply folds onto itself, and so that makes it express more quickly. So the final thing you have a choice or a decision to make with gene therapy is what your delivery route is going to be. And in this particular picture, it's just showing the heart specifically, but it really applies to the entire body. So you could deliver intravenously, and basically, in this example, cover the whole heart or deliver something throughout the entire body. You could choose vascular delivery through the coronary artery, which would really target specifically the heart. You could use intramyocardial or intramuscular delivery, which would require separate injections, probably multiple injections, into the actual meat of the muscle. So these are the vectors we've developed to really test AAV-mediated gene therapy for Barth syndrome. This example is showing the Desmond promoter, which I mentioned before, the CMV promoter, which is a very ubiquitous, really strong promoter. It would probably be stronger expression than the Desmond promoter, but it may also express tafazin in tissues we may not really need it to be expressed in. And then finally, there's not actually a known TAS promoter, but if you look at the upstream sequence of TAS, there are some potentially promoter-like elements, so we just kind of took a chunk of that and stuck it in front of TAS. And then for the purposes of most of these studies, which the promoter studies are being done in mice, I did not codon optimize this for humans to then test it in mice, so it's not codon optimized. But it does have a MIC tag to help facilitate tracking of the protein. And then finally, we've made different viruses. We've got AAV9, which would be used for the in vivo studies. And then this is an engineered capsid mutant, which really works well for in vitro studies. So it's kind of an interesting virology thing. Even things that target the heart well in vivo, they don't necessarily target cardiomyocytes well if you're doing in vitro experiments in a dish. So we play with the capsids to do what we need to do. So I'm just going to show you some very few experiments that we've done in vitro. One of the models we use are Barth patient-derived fibroblasts. We use AdMyoD, another gene delivery system, to deliver MyoD, which basically converts fibroblasts into a skeletal myotube. And so it just makes it a little bit more relevant to the disease of Barth syndrome, and it definitely gives the cells more mitochondria, and so it's a little bit of a better model for us to work with. We treat them with the mutated capsid form of AAV, and in this case it was with the DES promoter expressing TAS, and then we assessed them 1 week later. And we found good increase of TAS expression in all of the different lines that we added this to. We looked at mitochondrial membrane potential using TMRE uptake, which is one version of a way you can look at mitochondrial membrane potential, and we saw increased mitochondrial membrane potentials in the mitochondria of cells that were treated versus those that were untreated from the same lines. And then we looked at MLCL-CL ratios. And so on the left, you can see what, a healthy cell expressing tafazin on its own. In the middle, those are Barth cells that are expressing AAV, CMV driving just a marker gene, so it's just GFP, green fluorescent protein. So it's not therapeutic, and you see the increased MLCL-CL ratios that you'd expect. And then on the right-hand side, you see the therapeutic vector has knocked down the MLCL-CL ratios back to normal levels. So for the in vitro work, we're trying to wrap up this fibroblast study. We're looking at the other promoters. There are other experiments we're doing, and we're trying to finish that up. We've also started acquiring different iPS-derived cardiomyocytes or iPS lines that we're differentiating into cardiomyocytes. And the real value of this is that in the different, with our gene therapy, I'd really like to show that it's beneficial regardless of that background tafazin mutation. And so I think the iPS models are a really great way to do that. So a little about the mouse, just so everyone knows what version of the mouse model we're using. This is the DOX-inducible knockdown model, the shRNA mouse model. We mate, and then and they're not on DOX, and then as soon as we remove the males from the females, we put the females on DOX, and it's the DOX chow. And so with the way our experiment is set up, all of the mice received all the experimental mice received 1 × 10⁻13 vector genomes per kilogram of either the Desmond version of our vector or the CMV version of our vector. We either treated them as adults when they were 3 months of age intravenously via the jugular vein, or as neonates at 2 days of age intravenously via the temporal vein. And then we exposed them we waited until they were 5 months of age. So this was 2 months after the adult injections or 5 months after the neonatal injections. We exposed them to hypoxia for 1 week. So it was 10% oxygen. And we did this because we did a few pilot experiments where we were looking at force mechanics in different muscles of these animals. And we found that by exposing them to hypoxia, that was a good stress to kind of magnify their phenotype, just based on force mechanics measurements. It affects even wild-type mice are affected a little bit by hypoxia, but it's really an effect that's magnified in the TAS mice. And so I'd like to point out Michael Jones has a poster tonight, and he'd be glad to go into great detail about all the force mechanics studies that he's done. But very briefly, we're looking at the soleus muscle, which is kind of in the back of the hind limb of the animals. It's an ex vivo experiment. We pull the muscle out. The soleus is a slow twitch muscle. It uses a lot of oxidative phosphorylation for energy production, and it has lots of mitochondria in it, so it's very relevant to Barth syndrome. So we pull it out, and then this muscle gets hung here, and there are various experiments we can perform, and basically we're able to look at how fatigable this muscle is and how strong it is. And so this basically sums it up, whether they were treated as neonates or adults, whether they were treated with CMV or the Desmond constructs, all of them were indistinguishable from wild-type controls, whereas this line down here, the bottom line, is the TAS untreated animals. And so while TAS untreated animals are definitely more fatigable, we don't see that in any of the treatment groups. And then this is just another way of looking at that, basically looking at the last point on that previous graph. And so this is % max force, and you can see that there's a significant decrease in the % max force in the TAS animals as opposed to any of the treatment groups or wild-type controls, which were all statistically not significantly different from one another. Force frequency is more of a measure of strength, and again, all of the treatment groups as well as the wild-type controls are sort of interchangeable. They all look pretty much the same, whereas the untreated TAS mice had less strength. We did heart weight body weight ratios, and so these are in the adults, and the TAS mice that were untreated had significant increase in heart weight body weight ratios, whereas the treated did not. And then when we looked at those mice treated as neonates, we saw the exact same thing. So this is data hot off the press. A lot of these animals died last Friday. So There are other things I want to do with these tissues, and we just haven't had time to really assess them. But what I can say about the data is that the 2 promoters that we've tested thus far both appear to be functional based on just functional data. And no adverse events were observed, so all the animals tolerated the vectors well, regardless of when that was delivered or what they were given. We are obviously going to do all kinds of other experiments with these. We've got more mice being treated with all 3 promoters. We're going to do echocardiograms for non-invasive cardiac functional analyses, more of the MLCL analyses in the animals. We're ready to do EM analyses on these, and another poster plug, Manash Ramanathan can show you how in fibroblasts we assess mitochondrial structure. And then we really want to look at vector-derived transcription and translation. So I'd like to know, with this delivery dose, how much transcription is really coming from that delivery dose. And so these are some examples of some recent or within the last few years successful AAV gene therapy trials. I'm not going to go into detail of these, but by successful, I basically mean there were no adverse events associated with delivery of the therapeutic, and there was some sort of significant improvement in whatever the disorder is. And then the one really exciting thing for everyone in the gene therapy field is that the European Union's version of the FDA approved an AAV-mediated gene therapy and that's called Glybera. It's for lipoprotein lipase deficiency. So it's really never too early to start thinking about planning for a clinical trial, especially for gene therapy, because there's so much that goes into it ahead of time. And so even though we're very early in sort of assessing all of these data and running these experiments, we've developed a team, and so Barry Byrne and Todd Cade and I have started thinking about what needs to go into preparing if we were to do a gene therapy trial with this, given whatever vector X winds up being, what kinds of things need to happen? What would the tox study in mice need to look like? What would a potential tox study in non-human primates need to look like? What are the different outcome measures we'd want to look at and things like that? So we're thinking about that now, because if we wait until I figure out exactly which vector we want to use, it's just going to take longer for it to actually happen in the clinic. So I'd like to thank everyone who's been involved with this. These are people involved with it in my lab. These are a bunch of Barth collaborators that we have, and thanks very much to this foundation for the support through my grant. I've had my lab for just over 2 years now, and this has been really wonderful. So thank you for your support.
Thank you very much. That's extremely exciting, and even the preliminary results are very impressive. Any questions or comments?
Well, one question I get, because I have the mic, you said the mice died. You mean you sacrificed the mice,
Yeah.
right?
We sacrificed,
Okay.
yeah, we killed them.
Just wanted to make sure.
It's a good question.
Hi, Robin Duncan from Waterloo. Is there any insertion? Are these insertional vectors? And then in that case, there's been no insertion, no
It persists
spontaneous?
It's very rare.
Yep.
So yeah, and we've tried to detect it, and it's, you can't say never, but it's virtually undetectable, or it's very rare that it inserts into the genome. It really persists as an episome over time, and that's one of the reasons it persists for a long period of time.
People must be getting, oh, here's Sasha.
It's a quick question. Was there exon 5 present on this?
There was. So we do have an exon 5 deleted version of it, but we haven't gone to that. So this particular one has exon 5 in it. That's a good question. I don't have an explanation for it.